Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will discuss part 4 of the skill-based course computer-aided drug designing. So before wasting much more time, let's proceed what we have covered so far. In previous three notebooks or in previous 3 sessions, we have covered how we can identify our data in UniProt, search it in ChEMBL, then how can we download that data from those bioassays, how can we manage, and then finally we learned how to apply Lipinski rule on the whole dataset. In part 3 we learned how to clean that data from the unwanted substructures. Now in this section what we will do, we will perform a kind of virtual screening based on ligand similarity. So we will learn so much new things specifically in this notebook, and I'm sure many things will be new for you and you will be happy. So let's start. So just drop this code as I always do. And then come here to CADD project and select ligand similarity based on compound similarity. Connect. Okay. Once again, this is just a brief introduction. You can skip it. What are the aims of this tutorial? The aim of this tutorial is to learn how to perform ligand similarity. What does it mean? You need to understand it first. You assume that actually we have created some data set, and that is I think some 5,000 or 2,000 compound. That doesn't matter, but we have a data set which was created in previous parts. Now I assume that you must have some compounds from your lab, or you got it from some database like Zinc database or other PubChem or somewhere else. Now how to use this library of compounds which are already tested in the form of bioassays by different labs, how to use this information and extract those compounds which contain similarity to the ligands you have or you want to test it in docking. So for example, you have 10,000 compounds in that library, and here you have 10 compounds. So it will not be a good idea to just start docking. The good idea will be, or the perfect idea will be, to just what you need to do, just collect your library, 10 compounds, 20 compounds, or most of the people are using LCMS. I know that. So if you have compounds came from the LCMS, so why not match those compounds with that library and then select what are your potential compounds in this LCMS data, which will eventually can provide you a positive hit. No one can guarantee that they will provide you a positive hit, because this is a kind of predictive science. But the more you work with a proper methodology, the more you will have the higher chances of success you will have. So in this tutorial, we will use gefitinib. It's a drug against EGFR, and it is FDA approved, I guess. So we will use, let's say this is our drug, and we have the data on the back end, which we have prepared in the last 2 sessions. So how will we use this gefitinib, and how will we screen it against our own library? What you need to understand first in terms of theory, you need to understand about some fingerprints, and you need to understand about the measurement methods of those fingerprints. So there are 2 measurement methods. To the one we call it Tanimoto coefficient, and to the other we call it Dice coefficient. So we will compare our molecule, in this case gefitinib, to that library based on Tanimoto coefficient and based on Dice coefficient. But what we will compare, this is a question. For every comparison, if I talk to you just straightly, for every comparison everywhere, you first select some variables. From the variable, I means, if I tell you, like if I show you 2 different dresses, and if I tell you which one is more beautiful, or which one do you like, and you answer me the right one, the one in my right hand, then I can ask you why. So you will answer me that maybe the color is darker and I like the dark color, or maybe this dress has some unique logos or some unique prints. So what was that color and what was that prints? In short terms, in other terms, these were actually those 2 variables on which you have decided that which dress do you like. So the same way, if instead of dress we keep compounds, the same way, if we decide which compound is more similar to the drug we are trying to test, or I would say this way, we can also say the other way around, that which compounds in the library our potential drugs contain the similarity. So this similarity will be, we will call it fingerprints, or you can also say variables, but the correct word is a fingerprint. No, so let's learn about the details of the fingerprints. What actually are fingerprints? We have 3 kinds of fingerprints. We call it 1D, 2D and 3D. 1D fingerprints are normally the properties which we have already calculated in part 2, I guess, and that we call it Lipinski rules. So for Lipinski rule, what we have calculated, if you remember, we calculated molecular weight, we calculated log P and solubility and all those things, although we did not calculate melting point, but that's okay. So such kind of properties, in simple terms we call it 1D fingerprints. Now what are 2D fingerprints? from 2D fingerprints, it means that these fingerprints deals with the structure. Therefore, they are 2D. In simply, you need to remember this way. These fingerprints deals with the structure. So, what is the structure of a compound? You know, it's the combination of some aromatic rings like benzene or some octane or whatever, some carbon, hydrogen, some nitrogen, or maybe some oxygen, some fluorine, bromine. So the collection, and they all are connected by some lengths we call in chemistry bonds. So all the connection between these atoms makes a structure of a compound. If I ask you, if I give you two structures, just take it this way. If I give you two structures, one in your right hand and one in your left hand, and I tell you that compare these two structures for me, how much they are similar. So the easiest way is to broken down all these structures into their respective fragments. For example, if both the structure contains two benzene rings, then you can tell me these two are similar because both of them contains two benzene rings, both of them contain this functional group or this atom, etc., etc. So in this case, you are making decisions based on the structure, or I would say the fragments of structures, or the pieces of structure. So it means you are making decision based on the 2D fingerprints. What are the fingerprints? In simple terms, everyone, we have a fingerprints. So how these digital databases they recognize us from the patterns of these fingerprints. My patrons, maybe if I have 10 patrons, so 3 will be circular, 2 will be straighter, take it this way. Then the same way, these pieces of atoms which makes a molecular structure of a compound, they are somehow fingerprints like we have on our fingertips. I hope I clearly differentiated for you what are 1D descriptors and what are 2D descriptors. Let's move forward. So you remember these are the molecular descriptor. What we will do, we will calculate the molecular descriptors for the whole data set which we have, and we will also calculate the molecular descriptor for our compounds which we have in lab. In this case we have gefitinib. Okay, now next what we will do, I did not discuss the 3D molecular descriptor. The reason is because in this case we will not deal with the 3D molecular descriptor, therefore I skipped it. But in short, in 1 or 2 sentences, I can say like the molecular descriptor basically deals mostly with the conformation. Conformation means the exact orientation of those pieces of those functional groups in 3D space. And you know what are 3D space? It's a very vast topic. I can speak for more than hours on this topic, but I will not. But simply you take the understanding this way, like we have 3 axes. One is x axis, second is y axis, and the third one is z axis. So when we put the molecule somewhere, so maybe some part is straighter, some part is down towards the backward, some like bended towards the front, or up towards u, I would say, in the z-axis. So the point is, so all this makes the conformation. So if you deal compounds based on the conformation, such kind of coordinates representation of those pieces, of those fingerprints, then it makes the 3D molecular descriptor. But we will not deal 3D molecular descriptor in this notebook. Okay, so I hope I have covered for you that on what aspects we will make similarity decisions. But wait. Okay, what I explain here is now you will understand this. For these molecular fingerprints, or we also call it substructure based fingerprints, to the first set of fingerprints we call it MAX fingerprints, molecular access system fingerprints. Actually, what they did, they have calculated some notable fragments, some notable pieces, some notable key substructures, and they are defined. It means they are saved in the RDKit, and it's like this. You can see the structures is broken down into some pieces, like it has SS bond, it will have one benzene ring or some adiabatic ring, or it will contains fluorine, chlorine, bromine and iodine, and if it is present it will be colored, and if it is not present it will be white. But it's not a color and white. In computer language it will be 1 or 0. It means if it is 1, it means that both the compounds contain fluorine, chlorine, bromine and iodine. But if it is 0, it means absent. If it is, this box is 1 for the value is 1 for one compound and 0 for another compound, it means one compound contains these atoms but the other do not. So the computer read it this way. The second one is Morgan fingerprints. Actually, the Morgan fingerprints deals with the aromatic pieces of structures. Simply, like benzene ring is an aromatic and not a linear structure. So these information are also embedded in the RD kit, which we will use. Now move down. How we will compare, how we will measure such kind of similarities between compounds. What we will measure, we discussed in a quite sufficient detail. We will measure different kinds of fingerprints. Could be Max fingerprints or could be Morgan fingerprints, I will say this way. But how we will measure? So we will measure by two methods. The one is called Tanimoto coefficient and the other is called Dice coefficient. What is the difference between Tanimoto coefficient and Dice coefficient? According to the CCDC, Tanimoto is the number of features common to both molecules and the range is 0 to 1. If in the beginning I gave you the example of two dresses, let's come to that example again. For example, these two dresses are two structures. So how many features are common between these two dresses? For example, both the dresses are red color. If both the dresses are of red color, so this feature is common. But One contains circular prints and the other contains some linear prints. This feature is not common. So, coming to the point, the same way the Tanimoto coefficient will compare different structure. If they contain the same feature, it will say they are similar. If they contain a different feature, it will say different. This will be the formula the program will calculate. So A stands for number of features present in molecule A, B with the number of features present in molecule B, and how many features they both will share. Like in the dress example, I gave you a red feature is shared by the two dresses. Okay, coming to the dice coefficient, it is almost somehow similar, but according to the CCDC definition, the number of features common to both molecules, but it should be relative to the average size of the total number of features. Like how many total features are present between the two molecules, and then how many of them are shared by those two molecules. So this we will call it the dice coefficient, and this will be the formula. Normally the dice coefficient will give you the result will be higher than the in terms of values, it will generate a higher value than Tanimoto because of the encoded algorithm or encoded formula. Okay, what we will do some screening, and we will select some molecule. Let's say in this case give it a nib, we will try something. Okay, so and also finally we will do some enrichment plots to see whether how much similarity we are finding or we are not finding, whatever. So let's say, without wasting much more time, I have already discussed in a sufficient detail much more things. For further, I would say you can read it, it's okay. So let's say install, and then after installation if you are kind of hesitated from my explanation, so I told you in part first that I will dive in a proper teaching mood, and I also keep in mind that most of my listeners are from paper biology background, so I'm trying my best to make things easy for them and that they can digest in the easy way these kind of information. Okay, we installed the RDKit and we imported some libraries, the important libraries. Now here first we will test all these things on some virtual data. I mean, we will not directly start work on our own library of data compounds. We will just work on some compound. Let's say how many of them: 1 2 3 4 5 6 8. So these are the 8 smiley structure, and these are their names. We will run it so that the program records our program recorded this. Now we will draw these molecules. So these are those 8 molecules which we gave to the programs, and we just mentioned 2 of them. You can see molecular heads, so don't be confused that why 2, why not 8. If you want to make them 8, just change that 2 to 8. Now we will just try to show all of them. You can see these 8 molecules. Now let's say calculate molecular descriptors or fingerprints for them. So let me calculate first the 1D. In 1D I have calculated only the molecular weight, and now then I put all the molecular weights here, you can see. So for every compound I can see representation of molecular weight. Now if you want, you can draw these molecular weights. Now instead of name you can also see representation of molecular weight in this picture. Now based on the molecular weight, if you see these two compounds, doxycycline and tetracycline, they are almost similar. You see, almost 4 and almost the structure is also kind of similar with few exceptions here. So based on the 1D molecular, this is interesting, you need to pay attention. Based on the 1D molecular descriptor, these two compounds are similar. Now let's see, we will calculate molecular descriptors for 2D. And among 2D we have two. One was Morgan and one was Max. So let's say generate the Max fingerprints, and you can see 010101 because I told you in the start that computer read this way. So we will not understand, but the computer understand this. Okay, now we had created the Max fingerprints or the 2D. Now we will create the Morgan fingerprints. Not create, actually generate. Sorry, because English is not my mother language. Okay, again 001 something. So what we did, we calculate the 1D descriptor and we calculate the 2D descriptor, but the 2D descriptor are two different types. One is for those Morgan, which mainly deals with the aromatic, and one is for the Max. Now we will apply this to the molecule data frame, and let's say let's see what we get. Now we have our data now for the fingerprints which are calculated. Now we will apply the similarity measures. Now what are the similarity? It's just like that. I gave you an example for about like when I gave you two molecules in your hand, left and right hands, you just split them into pieces. You did not calculate it yet. So the computer just remembered all those fingerprints. Now we will calculate, let's say calculate the Tanimoto coefficient between two molecules. Okay, and that was some similarity came from 0.59, and we wanted to check the computer, like to check the code, that whether it's running okay or not, correctly or not. So here we calculate the similarity of the same molecule. So that is 1. It means 100% similar. Now what we did, we will now Max fingerprints, Tanimoto similarity, and we have calculated two kinds of fingerprints, Max and Morgan. Now we will check the max fingerprints on both the methods, Tanimoto and dice. You can see the doxycycline and the tetracycline and all these for the Tanimoto, these are the values, and for the dice these are the values. I told you that the dice will be slightly higher, but that doesn't matter because of the formula. Okay, based on the Tanimoto and dice similarity, we got almost the same kind of ranking. Now we will draw this similarity stuff. So based on Tanimoto, you can see doxycycline and tetracycline and all those values were fetched with the actual diagrams. Now if you see, with max fingerprints, tetracycline is most similar, high score, followed by amoxicillin. In contrast, with 1D descriptor, where we saw that the linear in 1D what we saw, if you come here again, in 1D doxycycline and tetracycline were almost similar, but here almost the result are still same, tetracycline and doxycycline, but if you compare both these figures, this aliphatic was somewhere here based on the molecular weight, but it comes out almost dissimilar in the max fingerprints. Now we will also calculate the same thing for Morgan. We compare the Tanimoto similarity based on Max and Morgan fingerprints. Therefore, we plot the Tanimoto similarity versus this. Let me plot. Okay. Actually, this data we are going to plot because this data is a bit different. If you can see doxycycline and here the tetracycline similarity level has been dropped 0.53, which was higher for the max. Why it has been dropped? Because if you remember, the Morgan similarity calculates the aromatic stuffs. Okay. This is a kind of matrix diagram, I would say. You can read it from any side you like, so therefore it's here. Okay, so these 7 or 8 compounds, they are plotted here, and the description is here. You can read here that what we did, we just compare the max and Morgan fingerprints based on the Tanimoto similarity, and the rankings were shown here. Morgan fingerprints also recognized tetracycline as the most similar compound to doxycycline, but with a low score. You can see this is 0.53. Okay, now it was just until now we just deal with variety of data. We just deal with the data, and now we will upload our real data to this notebook, and will calculate something real. So the above exercise was somehow for your understanding that how the Tanimoto work, how the Morgan work, the dice works for both those kind of fingerprints. Okay, let's upload the data. I will upload the data generated from part 2 RO5 properties filters. So the data is already present in my download folder. Its name is filtered 1 there, so I will write filter 1. Now we have all those 5004 and 30 compounds. Some of you can ask me that why you have uploaded this data, why not the clean data generated in part 3. Yeah, sure, you can upload that data, that doesn't matter. Okay, this is our query molecule. So are you getting now? This is the library, and this is the molecule which we want to test. It's a kind of virtual screening based on ligand similarity, or you can say this is the compound which came from LCMS if you have performed some LCMS data. Now here we have uploaded one molecule. You can upload it as many as you like. If you don't know how to write those, just put a comma here and then paste another one. Otherwise you can also read the first code where we have uploaded 8 molecules, so you can extend that code, that doesn't matter. Okay, and now generate Max and Morgan fingerprint for the query molecule. Query molecule is this one. Generate Max and Morgan fingerprints for the whole data set. Now it will take some time because it generates the fingerprints information for all those 5400 something is done. Calculate the Tanimoto similarity between the query molecule, which is gefitinib, and with our molecular data set. Calculate it. Now calculate the dice similarity based on the dice coefficient. Now print it here. So all the molecules, here you can see for this molecule Tanimoto is 0.4, Tanimoto max is 0.4, the Tanimoto Morgan is 0.3, the dice max is 0.5, and the dice Morgan is 0.4. Almost similar data. I just asked the program to print 3 molecules for me with this kind of result. Now I will want to see some distribution in a picture. Okay, so for data we have four: Tanimoto Max, Tanimoto Morgan, Dice Max, and Dice Morgan. I hope you understand now because we have spent sufficient detail in the start. So most of the compounds by Tanimoto Max, they lies in this range, zero point 0.4 to I would say 0.7 here. But for the Tanimoto Morgans, it says like the similarity threshold is a bit on the lower side, and the same way you can read this data. Second, we can compare the similarities here. This time let's directly compare Tanimoto and Dice similarity for the two fingerprints. This is for Tanimoto Max, this is for Tanimoto Morgan. Okay, so it just tells you this graph, like how much the value of how much is these compounds are similar to our compounds, query compound in fact, which is gefitinib. Now visualize the most similar compounds. We ask the program that okay, just give me the list of most similar compounds, but I ask for 3, so it will generate 3. You can ask for more if you are working with some real data. So you can see that the results on the higher side. So this way you can, this is an important step to understand. For example, you have some compounds, in this case we have gefitinib. Now I would suggest you make some threshold line, let's say 0.6 or 0.7. Please do not ask me what is the good threshold line. It depends on experiment. If I have so many compounds matching, then of course I will set a higher threshold line, otherwise I will drop down myself. So you need to select a threshold line and increase this number 3 to 4, 5 to 6 heads, whatever, and then you will know what are those compounds are to which your query compounds are matching, or is there anyone or not. This is also an important question. Now this is an important thing because the data almost contains the bioactivity in the form of PIC50 value we discussed in part 1. So the higher PIC50 value means better compound. In this case, this is our query compound, but from the ChEMBL, this compound, it shows the highest bioactivity data. It shows this. Okay, now you will understand now the exact theme of this notebook. It means this compound is a potential inhibitor or potential activator for whatever reason you are using, but remember IC50 is for inhibitor and EC value or the EC unit is somehow using for agonist or activator. So this is our potential, this is our query compound. It is matching with all those compounds which has a very higher bioactivity data from real life experiment. So it means you need to proceed this compound to your virtual screen. But if it is not matching here, it means skip it. Don't waste much more time on it. Now Some people can ask me now, or they may have a question that what if we have two compounds or three compounds in query data? Then the simplest answer is that you don't need to follow this procedure. But if you have a long list of compounds, let's say 1000 compounds, so why waste your time on 1000 compounds? Just make analysis and select those 100 or 50 from those 1000 compounds which are matching with this higher bioactivity data, and do your docking, and you will not complain about the GPU's time, because 50-60 docking is very easy. You can even perform an offline setup. And another beauty of this method is that if you perform all these stuffs, these calculations, of course your papers will go high, because you did not blind perform some kind of just pick compounds and docked it. No, you came to the docking step with a proper scientific procedure. What you removed, what you kept, why you removed and why you kept. This is the main question. Okay, now we will generate some, you please read this by yourself. I want to quickly wind it off. Okay, we will now generate some enrichment plots to validate our screening of the detected active molecules. And how we'll generate, just run this cell. It's almost the same cell we ran above. Now we will calculate some molecular similarity, and we will use some PIC 50 cutoff. It is up to you which cutoff you are using. Wait, I'm looking for the exact place to be used for the cutoff. Active counter plus -1, active counter plus. Okay, here it is. So what I told you about the threshold value, so in this experiment they will use 6.3. You can use 7, you can use 5, it is up to you. It means that do not include those compounds in enrichment analysis which are below 6.3. In your case, it will be your decision. Now prepare data by running these cells, and you will have this kind of stuff. Plot the enrichment data. Oh, it gave some error. I will check what error it says. No data is defined. Wait, let me fix it and come back. Okay, we have made some mistakes, so I will teach you here how, because this will also generate an error for you. If you see here at the end of this plot, it says save plot. So here this address is wrong. So what you need to write, you just if you see here, expand this panel and just copy something. Let's say the readme copy path. Now what you will need, just copy this way and leave the PNG as it is and say Control V. Now content, sample data, readme.md, just change it to some name like enrichment dot png. Now let me run again. Oh, what is the problem now? Oh, line 41 showed some error. Let's say, okay, let's do this way. I think I missed this. No. Put here, so it will change the color. It means now it will be fine now. So what you need to do, just copy the path, change the names, leave the png as it is, and put this double dash line here, and you will see it will change the color. Now it will be fine. Yes. What name we used? enrichment.png. So we should have this file here, enrichment.png. You can download it. That's it. Okay, is actually this figure, and true activities identified. So this is about the Meg's fingerprints. This orange kind of line is about the Morgan fingerprints. This is the optimal, and this is the random. So the random is just a line, it has no meaning. But the optimal, it means optimal is higher. You can see here. So what does it shows? It shows that the query compound we use, which was getFnib, that query compound actually it actually matches to the molecules. We use a cutoff of 6.3, so that actually matches to the molecules which shows higher PIC50 values. It means the active molecules. So to sum up, to make a conclusion for you, for example, you have 10 molecules in your query molecules, and if you test all those 10 molecules this way by this notebook, at the end you will have some data. The data will be either this way, this line will be on the higher side, or the data will be different. I mean, you may not find some good hits on similarity. So if you did not find good hits, I would suggest don't waste your time on those molecules and proceeding them to the docking setup. Just skip it, just remove them. So based on this tutorial, I hope you will be able now to screen your library of compounds which you have generated from anywhere, and to screen them for the potential compounds. Now dock those potential compounds, or the other way is like people are just picking compounds and then docked it. So that is wrong. Technically that is wrong, because maybe that contains some unwanted structures, superstructures, or some pans which we covered in section 2. Maybe they are avoiding Lipinski, which is not a hard and fast rule, which we covered in part 2. Maybe they do not contain some structure similarity. Now a question comes, what is the importance of structure similarity? The answer is that the active pocket, in this case EGFR, the EGFR of active pocket is same. I mean, it is not changing. So if it is same and there are too many bioassays available, let's say 5,000 compound in our case, so why not make a pre-guess or a smart guess that what from my library of compounds, what are those compounds which will be fitted in the active pocket of EGFR. So of course the higher probability is of those compounds which show similarity of the already tested compounds. I hope I have made things clear for you now, and you can also calculate some enrichment factor here. You can read this and possibly you observed actually it's so late with me and I'm recording this video, so that's why I'm somehow dropping my lines. Okay, so here we are calculating the enrichment factors. So in this script we calculate some enrichment factors, and if you read here it says that based on the above plot, the idea is to determine the percentage of the active molecules in the top 5%. So how will you determine? We can determine them from the enrichment factors. We will use some helper functions here, and once you click it, then the helper function will calculate the random enrichment factors of the active molecules here, and from here you can calculate the enrichment factors for the optimal factors for the optimal enrichment. Here one is random, one is optimal, and rank data set with the 5% cutoff because we want the top 5% compounds. Then click this and it gave us that like experimental enrichment factor for 5% of the rank data set. In Tanumutu that was 6.9%, and for Morgan that was for McCase 6.9, and for Morgan that was 7.5. We also compare the random and optimal enrichment factor. So the random if the data set, the 5% of the data set, you can say randomly it was belong to the active site, active molecules. It means like based on the random factor here, this line, and this one is for optimal. So this calculation means that our query molecule, in this case only one molecule, GetFNF, it enriched with 8.4% of the compounds. It's a huge data. I mean, we have used 5,000 compounds almost. So in every 100 it is enriched with 8 compounds or 8.4 compounds. So you can calculate. So it means the GetFNF could be a true or valuable compound or a true positive compound to be proceeded to the docking material, the docking methodologies or the docking pipelines. So you can share all these results. I have also provided the citations to you, and I'm sorry because I also feel the last 10 minutes was too rough. I am kind of sleepy, so that's why. But I hope you will get it. And still, if you have any question, please ask in the comment section. I will try my best to answer you as quickly as possible. Read this discussion section, and once again, sorry for the last 10 minutes. I'm also human, and at times I feel low on energy. Okay, thank you. Bye bye.